Welcome everyone. Yeah, I'm happy to see so many faces here. It's really nice to be back in sunny Southern California, out of the winter. Yeah, as Todd said, I'm the business director for Amphasys. Amphasys is a 5-year-old spin-off company that produces the instruments, but the technology has been in development for over 15 years by our founder. And today I would like to talk to you about different examples of how our customers are improving what our customers are doing with the instrument and how they're applying pollen viability. First, I would like to talk about why our current customers are investing in pollen quality analysis, then a quick overview over how the technology works, and to go to the main part of the talk, which will be use cases for customers doing. In breeding, the biggest effect is cost savings by improving crossing efficiencies, especially of difficult to cross plants, and breeding for lines with higher tolerances, for example, more heat tolerance. In production, we have customers that are using the instrument to increase seed yields and also to monitor the pollen supply chain to ensure that pollination always occurs with highly viable pollen. Because many of you are researchers, I would like to end with 2 research examples. One is the selection of plant protection agents and how pollen viability plays a role there. And the other is how we can also even look at the microspore maturation path. Our technology is called impedance flow cytometry, and it works with a microfluidic chip at its heart. And the best way to show how this works is with a short video. In this chip, we measure the electrical resistance. And the chip is in a black plastic holder. So this is the chip. And it's a sandwich that consists of 2 glass layers, 2 electrodes, and a polymer film with a microfluidic channel that the cells pass through. And when the cells pass through, they pass between 2 electrodes, and an electrical field is applied in the low megahertz ranges. And live cells, they can compensate for the changes in this alternating current. And that's how we can distinguish between live and dead cells. Or another way to show this is with this close-up animation of, this is the microfluidic channel, 2 pairs of electrodes, and here you see a cell passed from the left to the right. So as the signal is completely electrical, there's no optics in the system, there's no need for labeling or staining the cells. At very low frequencies, an instrument can measure at multiple frequencies. So at low frequencies, what we see is that the cell membranes, they can kind of compensate for the quick changes in the alternating current, and we get information of the cell as a whole. And at high frequencies, the cells or pollen can't compensate anymore, the cell membrane becomes permeable, and we get information about the inside of the cytoplasm, and this also helps us to detect diploidy of the cells. And so what does this mean for pollen? Here you can see 2 measurements, and the second one is a heat inactivated sample. So we can clearly distinguish between them. Each dot here is a single cell. And on the y-axis, we see an indication for the size of the particle that we looked at. And on the x-axis is what we normally call the viability axis. And with this technology, we can measure 10,000 cells in a minute, so much faster, more than before. We can give it an accurate measurement for viability. And the nice thing is it works for all pollen species. Just want to give a quick comparison to the traditional technologies that were used, so you asked for it. FDA staining relies on microscopy, so you can only look at like a limited number of cells. The sample preparation is complex and it does not work for all pollen species, and yeah, there are things that make it really operator dependent. Fortunately, our technology correlates very nicely with an R-square of 0.99 to the traditional method, but we can measure because we can measure significantly more cells, we get a much lower error and very fast results. And I think this is the most exciting because you can measure so many more cells, you can do projects on a completely different scale, you can do new projects that were impossible before. And what about the cells where you can't stain, where germination assays are used? So we also look quickly at germination analysis. It's also relying on microscopy and is quite operator dependent. And our technology correlates really nicely as well with germination assays as well. Again, here you can do more pollen with a lower error and get a faster result. So with that was the technology overview and how it compares to traditional methods. I would like to get into use cases. Yes?
One quick question. Is there any possibility of sorting with this instrument?
It's a really good question. In theory, we could build it, but it would be quite expensive, and we haven't found use cases where people would be prepared to pay for that, but yeah, at the moment not. So the first use case I want to go into is a quote from Amphasys R&D director. They started by implementing this technology very broadly in all of their breeding programs. And the effect that the implementation of really testing the pollen viability is that they could use, by using our instrument, they were able to increase their crossing efficiencies by over 30%, which is quite amazing and really saved them a lot of costs. The main reason, or the main two things that contributed to this efficiency gain were that they were optimizing the timing of the pollen harvest to ensure that they had the pollen with the highest viability. And the other thing was that before pollination, they were able to select the samples with the optimal pollen viability. Another example to improve crossing efficiencies is to look at pollen ploidy. For instance, seedless watermelon are triploid but sterile, and you need a diploid and a tetraploid parent line, but you really want to know with what pollen that you cross. And as you can see here, we can clearly distinguish with our instruments a population of like diploid pollen and tetraploid pollen. This allows a very effective quick check before pollination. And as you're all aware, global warming requires breeding for higher tolerances, and as pollen is one of the most heat-sensitive tissues, a selection for pollen with a higher viability at elevated temperature leads to a selection for higher heat tolerance. But we can also use pollen viability as a parameter in selection for other tolerances as like one of the factors. And this was an example at Biocrop Science. They were looking at wheat, and because wheat pollen's not very long-lived, they took the instrument into the field to minimize the effect of the aging of pollen. And with the instrument, they were able to analyze 100 samples in less than 4 hours and had thereby measurements immediately without the effect of aging. In seed production, people are looking to increase the male to female ratio, also to increase efficiencies and save cost. And the current method for pollen counting is pollen deposition analysis and then counting in a Coulter counter. And so we wanted to know how we compare to that. And as you can see here, our measurements nicely correlate with what you can get with a Coulter counter. But in addition to this, we can also measure pollen viability. Is anyone doing pollen deposition analysis? No. I was just wondering how important viability would be in this case. Is anyone working with sterile male, cytoplasmic male sterile lines? But yes, because we're measuring pollen viability, you can also use it to check for pollen sterility. And this is an example of two CMS lines. Young lines had quite a low pollen viability, and the control was highly viable. And then that was compared to an older line. And in the older line, in the control, the pollen viability was reduced. But one of the two CMS lines had increased pollen viability. So it's a good technology to check the status of CMS lines. But then we're also thinking, like, what do you do if you have, what happens if you pollinate with samples of such different viability? This is an example of tomato. So below a certain percentage of pollen viability, you get no fruits. Above a certain level, you get fruits, but they're small with limited seeds. And above a certain threshold, you get the biggest fruits and crop yields. Another example of why pollen viability measurement before pollination is important is this example from research at Syngenta, which shows that for some of those flower lines, the optimal pollination was not the pollen with the highest yield, but with a lower viability. So this is an effect that we've seen a couple of times, and we're not sure yet how reproducible this is. My last example from breeding, I would like to talk about the pollen supply chain. In these cases, customers are separating the males from the female plants. Pollen gets harvested in the flower, and then the timing of the pollen harvest needs to be optimized to ensure optimal pollen viability. Then the storage conditions need to be optimized and transport. Before pollination. Or, to quote Joop Lambach from ENSA again, one of the biggest advantages lies in the continuous monitoring of pollen quality at the production site. It helped them mainly to prevent production losses, and it was really important when working with contractors that get paid by yield. Now I would like to come to my last two examples, a little bit more research oriented. The first one is the application of pesticides before pollination. In this example, pollen viability was monitored continuously, and after an infection with thrips was detected, pesticide was applied, and as you can see here, in the 2 following weeks, the pollen viability was so low that you would not have been able to pollinate with that effectively. So what's that mean? That means that pollen viability can also be used to check when the optimal time is to apply pesticides before pollination, or you could use it to select plant protection agents that have less of an effect on the pollen.
Well, also for weed control, when you knock out weeds, it tell you now's the time to apply something. That's great. That's a really great slide right there.
Thank you. And then the last example is the determination of developmental stages in the microspore maturation path. The traditional method is Dapi staining, and you stain the nuclei, you can distinguish between the different stages of the development, but then these pollen, they're stained, you can't use them anymore for anything. And what we have seen with our technology is that you can clearly differentiate between the different stages of the microspore maturation path, and because our signal is electrical, you can just measure the pollen or the microspores, use them again, and optimize the timing of your double haploid induction. Specifically because the method is very fast, so you know when the right time is, and with that follow the entire microspore maturation path. And with that, to summarize these examples, so most of them were based on pollen viability. We showed that we could also do an approximation to pollen germination. We can look at pollen ploidy. We've seen that we can show the microspore maturation path and count pollen as well. So to summarize, it's a very fast method, allowing you to do projects on a different scale and to measure pollen viability much more frequently than you're currently doing. Because we're measuring so many samples, it's a very repeatable method. For those customers who work with stable pollen, we also offer an auto sampler so that you can automate the resuspension and loading of up to 192 samples. As I said, no labeling, no damage to the pollen, and it really works for all pollen species tested so far, and I think right now we're over 170 and have optimized conditions for over 170 different pollen species. And last but not least, our customers mainly are using the instrument in the lab, but we've also seen it being used in the greenhouse, and several people have taken it out to the field in various configurations. And with that, I would like to give you a quick overview over our current installed base. So as you can see, like a lot of seed companies have already started to adopt our technology, as well as research customers. And with that, I would like to thank you very much for your attention and hope that you can stay on for Brian and a demonstration, and happy to answer any questions you might have.
Can you just lay out the cost of the instrument and sample per sample cost, things of that nature?
The sample per sample cost, if you, one of those chips, let's say, you can use it for more than 1,000 samples, but conservatively said, like around 1,000 samples, and then the cost per analysis per chip is what is it, about between 30 and 50 cents.
Yes.
And the instrument itself?
The instrument itself runs for a little bit over $45,000.
And the
Initial purchase.
warranty cost?
Well, we give 1 year warranty, and then there's a service plan we can offer.
Depending on where we are, we have different contracts that include everything. Yeah.
All right, at this point, we have the instrument in the front. You're welcome to join and maybe squeeze a little bit in so you can actually see it. Unfortunately, we couldn't connect it to the PowerPoint, so in order to see something, you really have to be close by. We'd appreciate if you come by. We have some pollen, and maybe Brian, you can say what you're going to show.
Sure. So I am actually going to show some corn pound sticks.